Hello, my name is Matthew Buckley, and today I'm going to be going over my concept map for membranes, and it's quite lengthy, so here it is. So basically we're going over the plasma membrane, and there's just some basic things about it. So we're going to start with the different kinds of proteins inside the phospholipid bilayer. So we have a picture right here which kind of shows integral membrane proteins, peripheral lipid, and phospholipid as well. So we start with peripheral, which is membrane proteins that adhere temporarily to the biological membrane. Protein regulates cell signaling and other functions. Integral proteins, membranous proteins serve as cytoskeleton anchors to plasma membrane permanently attached to the biological protein. Lipid-linked membrane is associated protein are proteins located on the surface of the cell membrane that are covalently attached to the lipids within the cell. So then we kind of have a section here talking about additional proteins, which I'll just expand on by starting with proteoglycan glycosylated proteins, which have covalent attached highly anionic glycosaminoglycans and provide regulation of satellite cell growth and development. Here we have some more additional proteins, or one additional protein, which is the glycoprotein, which is a comprised protein and carbohydrate chain involved in structure, reproduction, immunity, hormones, and protection of cells, organisms, and of the cell. Also important for cross-linked cells and proteins. And then we have just a little subsection, glycolipid, made of carbohydrates usually found on the extracellular face of the eukaryotic cellular membrane, and function to maintain stability of the membrane and facilitate cell interaction. Here's a picture of a glycolipid and a glycoprotein. And these are kind of just grouped together because they're both correlated to each other. As you can see, here's your glycolipid and glycoprotein, but it is not a protein, it is a lipid. But then we have some more proteins, which are membrane proteins, and this is kind of just the three different types of membrane proteins. So membrane proteins are permanently attached to the membrane and move ions and large solutes across the membranes. So we have a picture here of different kinds of membrane proteins. So I'll just go into the definition of the anchor, enzyme, receptor, and we don't have the transporter. We have that one down here, but it is inclusive to the membrane proteins. So an anchor membrane protein is a binding site for extracellular molecules, and when signaling through this binding site, an intercellular process is activated. Enzyme membrane proteins can transform molecules into different forms. So energy is just an example of this, into proper chemical forms to store it in the ATP. Receptor membrane proteins are receptors used for mediating the process of cell activation, cell adhesion, and signaling pathways based on the outside of the cell. And then transporter membrane proteins are the movement of small molecules across the membrane, macromolecules. So that's the overview of the membrane proteins, and then we go into membrane channels. Complexes of membrane proteins are peptides that mediate passive transports of solutes through the formation of aqueous diffusion through aquaporins. Then we have protein-attached membrane-associated protein, which is part of or interact with biological membrane. Permanent part of the cell membrane allow hydrophilic molecules and pass through the cell membrane. And finally, we have monolayer-associated membrane protein, which is proteins situated within the cell or cytocell, and they interact with the inner layer of the lipid bilayer. So that is quite a bit of proteins incorporated in the phospholipid bilayer. There's a lot of variety there. But now we move into different transport methods of molecules inside and outside of the cell. So we start off with active transport, which is the process of moving molecules across a cellular membrane through the use of cellular energy used to accumulate glucose and amino acids. Then we have the channel-mediated passive transport, which is diffusion of an ion which is charged, and the total number of electrons doesn't equal its total number of protons. Given it a positive or negative charge, they move across the membrane through a water-filled protein channel. And finally, we have passive transport, which is the movement of solute from a region of high electrochemical potential to one side of the cell membrane and to the opposite. So that is a more negative reaction, which is a preferable energy movement within a cell. And then we move on to osmosis and simple diffusion. So osmosis is the spontaneous net movement of solvent molecules through a selectively permeable membrane from high water potential to a region of low water potential. And then we have simple diffusion, which is diffusion of a molecule that does not require assistance of membrane proteins. Kind of solutions used in this process are hypotonic solution, which is a solution containing fewer dissolved particles when compared to normal cells. Hypertonic solution is a solution containing higher concentration of dissolved particles compared to normal cells. And then finally we have isotonic solution, which is a solution that has the same salt concentration as cells. The interior of the cell is the same as the outside in regards to concentration of the solution. And here are some pictures of that. Then we go into ion channels, which are specialized proteins in the plasma membrane providing passageway for charged ions to cross through the membrane down the electrochemical gradient. Gradient ion channel is a group of transmembrane ion channel proteins which open to allow ions to pass through the membrane in the responsive binding of a chemical messenger. And this is kind of separate, but this is just talking about selective permeability, which is a regulator in the plasma membrane, allowing passage of some substances to prevent others from entering the cell. So that is it for kind of the transport methods. I guess we have the different gradients, which would be inclusive to that, so I can go ahead and talk about that before we move on. The electrical chemical gradient is a gradient of electrochemical potential driving an ion to move in a different direction. The concentration gradient occurs when the concentration of particles is higher in one area than another, resulting from the unequal distribution of ions. And then I have some pictures for that as well. And that is the conclusion of the different transport methods. So the plasma membrane is what we've come back to, which is the separator of the interior of the cell from the outside, and it is found in all cells. The phospholipid bilayer is what the cell membranes of most cells are made of, and it is a polar membrane made of lipids. Kind of just a side point here. A vesicle is a structure within or outside a cell made up of a liquid or cytoplasm enclosed by a lipid bilayer, formed from exocytosis or endocytosis, and they function as transports of materials that an organism needs to survive and recycle waste. So that's just something that is found in the phospholipid bilayer, which is a vesicle. And then we move on to different molecules that the cell or the phospholipid bilayer cells are made of. So we have detergent molecules, which are amphipathic molecules that contain polar or charged hydrophilic groups, functions to decrease surface tension of water from micelles. And then we have sterols and steroids, which are a subgroup of a hydroxyl group at the 3 position of the A ring. Main role is to provide stability. And then we kind of go back to the plasma membrane and then section off into what the plasma membrane is and what it is made of. So we have hydrophobic tail of phospholipid, which consists of a glycerol molecule, 2 fatty acids, and a phosphate group. They are uncharged, prevent phospholipid molecules from packing together to form a solid. So here we have a picture of the hydrophilic tail, which you can see is inside of it because they do not like water. And then we have the hydrophilic heads, which are outside, and then we have water here. Hydrophilic heads of the phospholipid are made up of a molecule containing the phosphate group and is negatively charged. They form bonds with water and other polar molecules. And these different tails are made up of saturated and unsaturated fats. So both of them have heads, and the only real difference here is double bonds in unsaturated fats and single bonds in saturated fats. So they're made up of carbon chains in saturated fats with no double bonds found in the phospholipid bilayer, and they're used to decrease bilayer fluidity. And then unsaturated fats made up of carbon chains with double bonds, also found in phospholipid bilayer, used to increase bilayer fluidity. So then we go back to the plasma membrane, and we have our two different ideas of what the phospholipid bilayer was. So we have the sandwich model, which was proposed by Davison and Daniele, and that is pretty much just a model of the cell membrane proposed by those two people in 1935, showing a hydrophilic and hydrophobic zone of the cell membrane. And here's a picture of what they kind of proposed. Then, in 1972, the fluid mosaic model was proposed by Singer and Nicholson, which shows proportions of proteins, lipids, and carbohydrates in the cell membrane. So kind of a little more information discovered about the phospholipid bilayer. And this is an amphipathic molecule, chemical compounds containing both polar and nonpolar portions in their structure. A phospholipid is an example of this. And that's just kind of, this picture is just showing you that the phospholipid bilayer is the structure, and it has a hydrophilic region and hydrophobic and polar and nonpolar region as displayed in the picture. Alrighty, well that is it. That is my concept map for the membrane, and thank you.